Hello everyone. I would like first to thank the organizers for giving me the opportunity to present my work. I am Musa Jibrin Ali from Chemical Engineering Department of Kocaeli University, Turkey. Today I'm going to present on the characterization of different photobioreactors for microalgae production. It is important to note that the whole work is being carried out under the supervision of Professor Bashar Uyar. The plan of the work is sectioned into 5 different parts. It starts by an introduction followed by objectives. We have materials and method, then the resultant discussion, and the whole will be railed in by a general conclusion. The idea of a photobioreactor has been around for a while. The first closed system showed up in the late 40s, but due to a lack of technical resources, their development was halted, and cultures were still being maintained in simple basins. In the 80s, research picked back up, and many photobioreactors were built and used in experiments with different geometric designs. The photobioreactor can be thought of as a closed system where biological interaction occur in the presence of light, energy, and which we attempt to control by adjusting the culture condition. The photobioreactor, which is the heart of the microalgae production, has a variety of geometrical forms, but constant property regardless of its use. From a geometrical perspective, two families of photobioreactor can be listed. We have flat photobioreactor and we have tubular photobioreactor. It is crucial to take into account that the geometry, dimension, volumes, culture thickness, illumination system, and the type of circulation before beginning a culture there. This study intends to determine the hydrodynamic, light distribution, and mass transfer features of these photobioreactors in addition to comparing how well they produce microalgae. After evaluating the outcomes, the best type will be selected. Rarely do research compare two bioreactors. Instead, the majority of recent studies operate many bioreactors simultaneously to assess their performance and show how they alter bioprocess. The simultaneous testing of 3 different species of large bioreactor make this work unique. As for materials and methods used in this study, here in this image we can see the complete set of the 3 different bioreactors that we installed in our laboratory. In the middle we have the stirred tank bioreactor with the stirrer placed on the top of it. On the left we have the bubble column bioreactor, and on the right we have internal loop airlift bioreactor. Results and discussions. In order to calculate the bubble size in the bioreactor, we use the software PaintNet to process the images of the bioreactor taken while working. By the help of this software, we can calculate the pixel square of every randomly selected bubble. Then we deduce the bubble diameter. For the gas holdup, we use a ruler to calculate the level difference of the liquid before and after aeration. This obtained difference can help us determine the gas holdup inside the bioreactor. The mixing time can be obtained by injecting the blue food dye in the reactor, with prealably working under a settled parameter. The difference between the start of the injection time and the homogeneity time is called mixing time. These 3 bar charts describe the bubble size distribution in the 3 bioreactors under the quite similar condition according to the type of the bioreactor. They are the expression of the number of bubbles versus the size range in every single bioreactor. At the first sight, it can easily be noticed that the 2 first bar charts of the airlift reactor and the stirred tank reactor are quite similar in terms of bubble distribution. And besides, in the both first bar charts, the dominant bubble size range is comprised between 2 to 3 mm. Meanwhile, in the bubble column bioreactor, the dominant bubble size range is 1.5 to 2. And this difference can be explained by the fact that in the 2 first reactor, the used disperser is a single-hole tube type disperser. Meanwhile, in the bubble column bioreactor, the used disperser is a multi-porous type. As the bubble size is the direct expression of the disperser orifice size, that's why we notice this difference. And also note that the internal tube of the airlift and the stirred presence in the stirred tank play a big role too. This result is expected because it is logic. In these graphs, we can see the effect of aspect ratio and gas rate on the gas holdup in the bioreactors. Based on these two graphs, we notice that the more the gas rate increases, the higher the gas holdup is. And in the contrary, the bigger the aspect ratio is, the smaller the gas holdup inside the reactor. And this is normal. The volumetric mass transfer coefficient details how efficient oxygen is transferred from the gas bubbles into the bioreactor medium. These curves are the representation of the time versus logarithmic concentration difference of the three bioreactors, and the linear part of the curve slope represents the KLA. The fact that the contact area of the bubble column bioreactor is bigger explains the high value of KLA in this bioreactor. The mixing time can be defined as the time needed to obtain a specific degree of homogeneity, and it is correlated with the specific power input and the geometrical parameters of the bioreactor. We used a Bluetooth dye to measure the mixing time by recording a video from the adding of the dye until the desired homogeneity. Then we processed the obtained images in the software called Color Max, which is a color analysis software built by my supervisor. It helped us to collect data and produce the mixing time curves. It is important to note that the previously obtained result is concerning the physical parameters of the three different photobioreactors. But right now, as you can see in this image, we are trying to grow microalgae inside them. The growth of the microalgae will be observed during days. Then, subsequently, we will collect data and compare the results and ended up selecting the optimal one. To conclude our presentation, at this level of study, we can say that bioreactors complement one another since every bioreactor has its own pros and cons. From the obtained results, we notice that the bubble column bioreactor performed very well in terms of oxygen mass transfer. But the problem with the bubble column is that it has a long mixing time and too much backmix while scaling it. And the other advantage of bubble column bioreactor is that it is the most affordable on the market, followed by airlift reactor. But the stirred tank bioreactor has a high power costs associated with impeller scaling. Thank you for your kind attention.